Welcome once again to BioPhorum's CGT Validation Challenges webinar. I hope you can all hear me. And today we will be hearing from Sharif Ahmed on the activity from CGT Validation Challenges group from BioPhorum. And what I will first do, however, is I will take you through a little background on BioPhorum before we get to the main focus of today's session from Sharif, for those members who may not be familiar with BioPhorum. So, BioPhorum is a global collaboration that brings together experts in the field like yourselves, where you can collaborate together, where you can share and identify common challenges and problems that you would like to discuss as a group of experts, and as a collaborative group from the wide industry, lots of different companies, you can get together, identify best practices and procedures to solve these common problems. And then within this collaboration, sharing experience, building networks, you can hopefully accelerate the development and improvements for the industry as a whole and have better, faster treatment to our patients. As you can see from this slide, BioPhorum has a large number of members, over 135 companies with over 6,000 subject matter experts like yourselves collaborating on a wide range of topics. So these topics are currently broken up into 9 forums or high-level topic areas that you can see here on the right. Now, each of these forums or high-level topic areas has a large number of specific sub-teams and working groups that are working towards specific topics. Now, the development group, drug substance, fill finish, information technology, med tech, supply partner, sustainability and newest forum, technology road mapping, and cell and gene therapy make up the whole BioPhorum. Now, the majority of these forums are focusing on mAbs, general biologics, small proteins, with the cell and gene therapy forum, as the name implies, focusing on all of these aspects covered by the others, but in the cell and gene therapy area. Now, some of these other forums may have the occasional cell and gene therapy focused topic, but they work closely with the CGT group to make sure that we get the correct experts and conversations going to deliver on what our members would like to discuss. Now, the cell and gene therapy group is almost 4 years old now, and that is what you'll be hearing from. This is what you'll be hearing from today about one of the teams, the Validation Challenges team. And during these 4 years, we have worked on a variety of topics. As you can see from these numbers, we have 43 members and company members and over 800 active participants, with Sharif, who you'll hear from in a moment, being one highly active member. During this time, all of the work the team have done have resulted in 22 publications and presentations. Now, these 7 work streams that you can see on your screen here cover a wide range of challenges within the cell and gene therapy space: commercialization, manufacturing challenges, environmental health and safety, analytics, raw materials, regulatory, validation, which will be today's focus, and also our newest work stream, RNA. So we're focusing here on all the RNA therapies, vaccines, and using it as a gene editing tool challenges. So each of these are working on a variety of topics, and between these 7 work streams, we have 28 other sub-teams below these working on dedicated topics, working towards deliverables decided by our members. Now, one of these sub-teams was the cell and gene therapy validation challenges topic, which is why you're here today on the call. And before I hand you over to Sharif, who will take you through this work and activity, I would like to introduce Sharif. So Sharif is an accomplished leader with the biotech industry with over 10 years experience. He's led a wide range of successful validation activities in therapeutic proteins, but most importantly for today's session, gene therapy manufacturing platforms. As you can see, He's graduated from the University of Utah in biology, has a PhD from Michigan State in genetics, and has postdoctoral work at Berkeley. Following his academic experience, he's worked in biorad laboratories, BioMarin, and most recently Bayer, where he has successfully led a wide variety of activities, including the CMC, quality scale-up, and process validation. As it shows, he is currently the lead for the supply of a recombinant factor VIII portfolio of products at Bayer, and he has been an active and great member of BioPhorum since 2016, and one of the founding members within the cell and gene therapy group as well. So before I hand over, I would just like to highlight Sharif will be taking you through these slides, but it's hopefully going to be an active discussion, and we welcome questions. And if you have any questions, please put them in the chat and we will look at them and we will address them towards the end of the discussion, but we really would welcome your questions. So lastly, all I'd like to do is to thank Sharif for agreeing to give this presentation and hand over to Sharif. So thank you very much, Sharif, and it's all over to you.
Thank you, Stephen, for the introduction as well as describing our efforts in BioPhorum. One request to either you or Nettie, I see these messages popping up about folks waiting in the waiting room. If there's any way for me to turn that off, because it can be a little distracting. But other than that, I really want to thank everybody. There is a large number of people who have joined the call, and I appreciate the interest. Really, I'm hoping that we'll have a good discussion around this topic. Today, we're going to talk about the cell and gene therapy validation challenges, which was an effort that was part of the cell and gene therapy work stream. And within this sub-team, we came together, and as the slide shows, there are several of us who were instrumental in putting this white paper together, in identifying some of the cell and gene therapy validation challenges. And we had, as you can see, a large number of supporting authors who also contributed from their experiences with the products that they are working on, and identifying some key challenges as well as offering some potential solutions. Next slide, please. Forward, there we go. The purpose, again, for the white paper was to highlight some of these challenges in validating cell and gene therapy, drug substance and drug product manufacturing processes, as well as analytical test methods. And we selected some challenges that were associated with validation of viral vector-mediated gene therapy, as well as gene-modified cell therapy manufacturing processes, and the associated analytical methods that we have encountered several challenges with, and offered some potential solutions for these. The aim for the sub-team in putting this paper together was to bring awareness of the complexity and challenges that are associated with validating cell and gene therapy manufacturing processes, in order to gain some industry-wide consensus in addressing some of these as early on in the processes as we can, and with the hope that this will help drive regulatory acceptance for a clear and fast path for successful validation for analytical methods as well as manufacturing processes. Next slide, please. As many of you know, cell and gene therapy products are a class of advanced therapeutics, and which offer tremendous potential. If I'm not mistaken, there are hundreds of cell and gene therapy products that are currently in development. The FDA has a large list of these products that are either going through clinical trials or are under review within the FDA. So although there's a tremendous potential in these advanced therapeutics, their manufacturing also offers a lot of challenges, and these tend to be much more complex than typical antibody or therapeutic protein products. Therefore, successful commercialization would require a multidisciplinary approach where one would have to integrate patient needs with our product, with the understanding and knowledge of the product, as well as our capability to manufacture these products. So for gene therapy, it typically requires modification of the expression of an individual's gene or repair an abnormal gene, where the DNA or RNA carrying the corrected or modified copy of the gene would be transferred to the somatic cells of a patient, and that hopefully would result in expression of a protein that has either therapeutic or curative value. On the other hand, for cell therapy, you're delivering live whole cells to the patient for the treatment of a given disease. And oftentimes, there is a particular gene of interest that is modified for its expression in case of gene-modified cell therapy, or altered biological properties of some living cells that are collected from either an autologous source, which is the same individual that you're going to return the therapy to, or an allogeneic source where another individual individual cells are used to modify and then deliver to a different patient. Next slide, please, Stephen. Within the white paper, we have compiled an existing list of guidance documents and presentation from recent that were recently published from global health authorities such as FDA, EMA, Health Canada, PMDA, and so on, and other references and publications at various conferences. And these are listed as within the appendix of the paper. And in this slide, you can see there's just a snapshot of the FDA guidance document that have come out in the last 10 some years now around various topics related to cell and gene therapy, its manufacturing, and the expectations that the agencies have. So I really encourage all of you to look through this list and individually deep dive into any of these regulations because they do offer a lot of insight into the thinking of health authorities and how they are, or what they are expecting in terms of programs that, specifically for cell and gene therapy. And these, expectations were definitely played a major role in developing these potential solutions at each one of our companies and as a team when we discuss these within BioPhorum sub-team. Next one, please. And what we also recognize is that as we started to identify some of these validation challenges in cell and gene therapy is that overall, the general process validation concepts and practices are applicable to all cell and gene therapy manufacturing processes and analytical methods. However, there are specific ones that are related to cell and gene therapy, and these continue to evolve. And as we get, your allogeneic or autologous cell therapies, there are some pluripotent, induced pluripotent stem cell therapy processes that are evolving, and they seem to have a specific need for that particular process. And some of these that we've identified are listed here in this slide, and of which we focus for this particular effort in this white paper on those challenges that are highlighted here in blue. And these are the challenges that I will dive a little deeper into in the subsequent slides as we go through this webinar. I would encourage everybody to read through the white paper and familiarize yourself for these other areas, which I'm not going to be covering today. There's some high-level discussions around these other topics. And as Stephen will show, that we continue to move forward with these other topics in the near future, and hope that they will also result in some solutions that we can provide for these important and areas of validation challenges that we have identified. Next slide, please, Stephen. So among the challenges that we discussed within the white paper, one of the first ones that I'd like to highlight here is related to the limited number of batches at commercial scale prior to and during PPQ. Now, for gene therapy processes, what we have found is that within our team of SMEs that represent organizations that have developed gene therapy or are in the process of developing gene therapy processes, that there's an opportunity to leverage platform processes. Gene therapy manufacturing process or viral vector manufacturing processes typically tend to can be leveraged from one product to another, whereas cell therapy products, as you may already know, are very much individual process and product driven. So for gene therapy products, we recommend that one uses or leverages platform processes that are available for within the organization or within the industry, and certainly use process and product information from similar products. So if you have one product that you've already developed and are in the process of commercializing that is utilizing an AAV system, you can certainly leverage that process and then utilize appropriately based on the product that you're developing for the next one. And the analytical tools that you develop can also help define your critical quality attributes, as well as designing an appropriate process control strategy. And our recommendations are around using DOEs at small scale. When you are using DOEs at small scale, we highly recommend to use material that is representative of the commercial manufacturing process. And I think this is a critical input and recommendation from the group, because you tend to see more variability in cell and gene therapy products as starting materials than you do typical antibody or therapeutic proteins. So utilizing those commercial material that is representative of commercial manufacturing process is very important. For cell therapy processes, there's additional challenge where many of the commercial manufacturing batches are at small scale. They're not your typical large-scale antibody manufacturing process where you're going into hundreds or thousands of liters of cell culture, for example. So this offers some unique challenges for validation purposes. And to circumvent some of these challenges, we recommend using healthy donor cells, as you may have patient cells that are limited in their availability. And also, we identified that the product may behave differently between healthy and diseased patient cells. So therefore, assessing the CQAs with healthy versus patient cell is one of the key recommendations that we offered, where understanding what really differentiates the disease versus the healthy cells and how to assess appropriate CQAs for them is really important. And then we have bridging studies as another potential solution and recommendation that we make together with risk assessments, which can then be used to justify the use of donor cells in your validation approach. And that should be, that should go hand in hand with developing analytical test methods that you have early on when you're using healthy cells, for example, or diseased cells, and ensure that you understand what appropriate process controls need to be in place. And when you're making process changes during your commercialization, going from clinical to a commercial setting, ensure that you have an analytical comparability plan that allows you to bridge the processes that are in the clinic with those that you are commercializing. Next one, please. So other challenges and potential solutions that we highlight in the paper include process characterization, which is critical to generating data to support process validation. Given the limited availability of material, as well as the small patient population size, it becomes very, very critical to have detailed and well-characterized process and generate that data that you can later on leverage your process validation activities. And this is where using material that is representative of commercial manufacturing process, for example, becomes very critical. Understanding and having that bridging of analytical method becomes very important, because you can then leverage a lot of that data that you've generated during process characterization, which remains very much relevant to your commercial practice and allow you to leverage for process validation downstream. And the limited supply of starting material, one of the recommendations that the team makes is around using surrogate materials. We all know how precious many of the cell therapy processes, for example, the material that is used for cell therapy manufacturing processes, are very much limited in supply. So when appropriate, use surrogate materials and use test samples from the actual starting materials to identify, especially for critical and key process steps. And the use of the justification for the use of surrogates is very much very critical to be able to use such surrogate material for the validation activities. And comparing and documenting how they represent your process stream and understand if they're, what the impact of any differences that you see are. And that is a very important aspect in addressing the use of materials. And further, material evaluation and characterization is also important for us to understand criticality and variability. And concurrent validation seems to be a very, very common strategy. And again, is very much hinged on the amount of process characterization that was done up front and the relevance of that process characterization work that allows you to support a concurrent validation. When you have limited supply of the starting materials and process intermediate, because there is an expected strong benefit to risk ratio for the patient. And then appropriate testing before using your cell bank for production of virus banks is an area that we touch upon together with purification of viral capsids and make some recommendations there. And finally, stage 3 process validation, or CPV, that is impacted by the small data set that is typically available for cell and gene therapy products. And what we recommend is to have a routine monitoring of your in-process and final release testing data of manufactured batches through the CPV program as you post-commercialization, to confirm that your the ability that you have for the to control the manufacturing process in manufacturing product that meets your product safety and quality requirements. So these are some of the additional challenges and potential solutions that we offer, and I will switch on to the next slide where we have a section on designing specifically designing PPQs for autologous cell therapy. Primarily because we see there's a high degree of variability between patient to patient within the autologous cell therapy processes, and the material that we have, the starting material that we have to initiate the manufacturing process is typically very limited in quantity. And typically, when the material that is generated from PPQ is not used in commercial setting. PPQ runs may be conducted using either healthy donor cells or patient cells, or a combination that is based on the way that you designed your study. And utilizing healthy donor cells as the starting material, where you're going to be using the same manufacturing process, is can be an option. And testing with the same methods that are going to be used for patient cells is very important again. Ensuring that you understand if there are any variabilities, where they are coming from, before you go into PPQs. And since healthy donor cells don't need to be, healthy donors don't need to be infused back with the final drug product, there is also that opportunity where you could use the material available at the end of a PPQ, for example, or during PPQ for extended testing, which is expected for PPQ. So in this kind of a situation, one could use healthy donor cells and perform those additional testing and understand how they now relate to the patient cells. And that could be a potential recommendation to go around this limited supply that is typically the case. And using data from your development studies, again, or based on manufacturing data that's available from clinical studies, is another recommendation. And understanding the systemic differences or biases that you see between two materials can be used to assess and justify the impact of any differences that you see. Material availability constraints are also one of the challenges that we discuss, where there's a high product variability due to variability in our starting material, especially in the case of patient cells. So understanding that variability is important. And then finally, it's important to establish appropriate acceptance criteria for PPQ runs so that you can account for that material variability. So we recommend that you, especially for cell therapy and to a great extent for gene therapy as well, that material differences or variability in material quality or material sources can be a major factor. So as you decide or as you establish your acceptance criteria for successful PPQ runs, ensure that you account for that material variability, which can go a long way. Next slide, please. And I think this may be one of the last slides where there are two analytical method validation challenges that we identify around assay variability and reference standards. Cell and gene therapy product testing typically uses specialized techniques, and there are instruments that are quite unique now that have not been quite as much used in antibody or therapeutic protein production or product testing. And these there seems to be also high assay variability that is often encountered. And again, given the limited amount of material availability for testing purposes, it also limits the opportunity that you can test and develop and improve on your method, the assays that are used to test your product. So as you develop the assay, as you validate the assay, it is key to understand the variability that comes that is inherent within the assay, or limitations or variability coming from operators or within the instruments. And using a risk-based, phase-appropriate analytical method validation strategy is important in this respect. And also, one recommendation that the team had was around trending your reference standard or assay control early on during method validation, method development prior to validation, so that you can understand how, as the method evolves, what are the sources and where are these sources of variability coming during your method development, which can be very, very useful and critical in the future as you go into method validation. And initially, we recommend using wider acceptance ranges. And as you optimize the process or optimize the assay, you can then establish acceptance ranges that will reflect these changes and improvements that you've made. In performing intra and inter-assay studies early in the life cycle is also another highly recommended approach to understand the variation that you see and where those variations are being contributed from. And maintaining frequent dialogue with the health agencies early on to keep them informed about your method development, about some of the challenges that you are facing throughout this development for guidance and acceptability is one of the most critical aspects of cell and gene therapy method development and validation. It gives you an opportunity to understand what health authorities are expecting, what are they considering to be appropriate, and how can you then either improve or enhance your the assay and reduce variations. And finally, and the last topic that I wanted to touch here today is around reference standards. And for cell and gene therapy manufacturing process, we highly recommend for you to use material that has been well characterized and is suitable to test identity, strength, potency, and purity of your product. Small number of reference standards for cell and gene therapy are currently available, and many of them have patient-specific variability. Therefore, we highly recommend that you establish and characterize in-house reference standards and develop and characterize and implement these assay controls so that you have appropriate reference standards available, which also allows you to understand the variability that has been contributed by the various sources, including your material. I think that's all I wanted to highlight within the paper. This was a wonderful experience from my end, and I'm sure all the authors would concur with me on that. Thank you very much for your attention, and I think Stephen and I will take any questions.
Yes, first of all, I'd like to thank you, Sharif, for presenting this and highlighting all of the great discussions. And before we get into the question section, I have shared earlier, but I'll share it again. In your Teams chat, just come is the link to the white paper that Sharif was just presenting on. So if you follow that, it will take you to the BioPhorum website where you can download the paper. You may need to create your own login to do this, but it's free to create a login. So please go and read all the extra information behind what Sharif has presented. So thank you, Sharif. And we have had a number of great questions coming in, and I will get to those in a moment. But I know you did ask, just before we take the questions, you wanted to highlight and also indicate why this topic was a priority to you and the team, and how the group kind of came up with these few, because there's lots of other validation challenges. Like, how did you choose these few challenges?
So I think part of it was certainly a team decision, and we used various different approaches in coming to that list that we start. That was an initial list that we felt like that we could start to work on. And we started out with a survey within our team members and collected the feedback from the respondents, and then took the ones that had the highest priority within the team. And it was a very well represented team coming from various companies, and then we saw that this gave an opportunity for us to cover a wide range from analytical methods to PPQs to material variability and so on. So I think certainly there was a process that was involved in collecting the feedback from the team on what everybody considered to be important to their organization, and then came down with what is available that we can at this time start to make some recommendations, because we have approached or come to that stage. So for example, if we're talking about PPQ experiences, what did we do with small batch sizes, or if you had limited amount, what could you use? How can you use surrogate materials? So obviously those recommendations had to come from experience that some of us within the team already had, that they could offer some recommendations that were acceptable to health authority and for licensure.
Great. Well, thank you for explaining that, Sharif. Now I'm just going to go through the questions, but please keep adding them and we'll try and get to your question. Now, the first question which we received was from Ramesh. Thank you. But I believe we may have already covered it. It's asking, can we use surrogate materials for the process and method validation in the case of personalized medicines? And I feel we have covered that. Would you say that is true, Sharif?
Yes, and I think I would recommend that you review the white paper as well as the presentation. And surrogate materials, again, you can use, but with that with that understanding that it's very important to justify and document as to why is it that you can use that surrogate material. And that includes, demonstration and documentation that the similarities that you have, the differences that you have, and how does that difference, if it impacts the outcome of that use.
Great. Thank you. I hope that helped, Ramesh. And just for the interest of time, got lots of questions. I'll move on. But as Sri said, please look at the paper as well. Benson has a kind of follow-up to that. Has anybody had used surrogate material for filter validation studies? So it requires relatively large volumes. I'll first of all put that to you, Sharif, but please, if anybody else is on the call, this is one of the real benefits of in these BioPhorum discussions. We have over 200 people here. So if you have something to add as well, please just put your hand up and we can hear your voice as well. But firstly, Sharif.
Yes, I mean, I have successfully used surrogate materials for filter validation, especially if you can demonstrate, for example, if you're, in the final stages of your final drug product filter validation, there are strategies that one can use, either, surrogate material, or you can have a modified version of your product where you can demonstrate that the modified version does not impact the outcome of the validation. And that's where I think it's very important to demonstrate what are the critical process parameters, what are the critical quality attributes for that particular step, for that process step, and how do these differ between the surrogate and your actual product. And if you can demonstrate that and document that, it is, in my experience, has been very well received by health agencies.
Thank you again. So the next question from Satish considering the fact that there is going to be definite variations in the starting material, apheresis, how will the CMA and CPP be controlled or established?
So again, I think this is where you know, there is not a lot of difference in how you, and the need for defining CMAs or the need for defining, you know, CQAs. In terms of how do you establish that, that will certainly be dependent on your understanding of the product as well as the process and those intermediates. So if you're starting out with, you know, patients, let's say, as the starting material, and you know from one patient to another patient, there is going to be difference, and that's what is required during process characterization. Understanding your process, understanding those starting materials and the sources of variability and thoroughly, and then again during your development studies, utilizing those, and then during PPQ, using healthy donor cells, and then having that appropriate connections between CQAs and CMAs is going to be very important.
Okay, thank you. And you mentioned healthy donor cells, and I know the most recent question we had is, can you share examples of surrogate material? Would that be the healthy donor cells?
So for surrogate materials, you can use, for example, if you have limited amount of you know, material in your drug product. I mean, one example that come to mind that we discussed within the team was for a gene therapy product where you could not use an entire batch of your viral vector gene therapy product through a, you know, for a filter validation study. So utilizing a surrogate solution, for example, where you have, you know, certain components of that of your final product being reduced, however, demonstrating that that reduction or changes in that composition is not going to be impacting the outcome of the validation, which in this case is essentially sterile filter validation. So typically you have, you know, when you're validating a filter, a sterile filter filtration process or the filter itself, you're looking at your ability to retain microbes, as well as also ensuring that you don't have absorption or loss of your product while you're going through that filtration step. So playing around with those conditions and then coming up with a potential surrogate material that allows you to maintain both of those aspects of that process step is going to be important. Unfortunately, I can't go into too much detail, but I think when you you know, think about what are the components of your final formulated drug product that is going to be passing through a sterile filter, what is the purpose of that step, and what do you expect there to be impacted? I think those will help you identify a potential surrogate material.
Great. Thank you, Sharif. Next question. We have lots coming, so I'll try and get through as many as possible. Do you prefer to have scaled down model developed before utilizing DOE for process characterization?
It's often useful to have scaled down models available as soon as you can. Now, scaled down models also have different scales. So depending on your availability of material as well as resources and time, one would expect that the closer you are with in terms of the use of material or in terms of scale, and you have a correlation between you and how scalable the process is, that will drive you know, what you can use for DOEs. But DOEs can be, you know, have been done at very small scale because of the need for a lot of material, a lot of number of you know, combinations of the parameters that you're trying to assess. So I think the closer you can be to a scaled down model that represents your commercial process would be the ideal.
Great, another great response. So from Anju, Can you comment on some of the extended characterization proposed for PPQ batches?
Extended. I would have to, Stephen, refer this to
Yes.
Wei for him to address that.
Okay. Apologies on that one, but again, possibly have a look in the article. It might be covered in a little bit more depth of the article. And again, that is something that unfortunately that lead isn't on today's call. But thank you for the question. And
if I may add here, Stephen, I think there are a certain set of extended characterization that are recommended in guidance documents that we've listed from the various health agencies, but it also is dependent on the program itself, the product itself. Each product for cell and gene therapy is going to be different. So it's going to be difficult for anybody to say what is going to be appropriate for your product, but I think you need to have read through those. I would recommend reading through the article as well as those guidance documents that highlight some that are your typical ones around safety, efficacy, and identity, and so on. But then additional ones would certainly be applicable based on what the product type is, and those would have to be negotiated with the health agencies.
okay, thank you for helping with that one. Another interesting question from Eric: is the use of only healthy donor material without any use of subject PPQ batches truly an acceptable PPQ approach? And has anyone done this with positive feedback?
I'm not 100% sure. I mean, I'm not sure, or I'm not aware of something similar to that situation, but a combination I have seen successfully being implemented. And I think that would be the more recommended one at this time. I would imagine that, certainly having a lot of development data and clinical data with patient cells would certainly bolster your case when you're using healthy donor cells for PPQ. But the key, again, is to demonstrate that relevance and demonstrate that you understand why the patient cells are different from healthy donor cells, and why when you go into commercial manufacturing using PPQs that runs that were done with healthy donor cells still allows you to produce a product that can go back to a patient.
Great, thank you. And I think you've kind of covered the next question from Satish Dara. What is the regulatory review on performing process validation with healthy donors and routine manufacture? And it's speaking with the agencies, and it's on a case by case. And also, I see that Humberto replied to that as well in the chat. So thank you. The agency is willing and happy to provide feedback during your discussions with all of these aspects around validation strategies. So that's really good.
Yes,
we have a question from Anand on specifically comment on an assay for sterility testing under CGT. Now, I don't believe we specifically in our group discussed sterility testing. However, I can say within one of the other groups within the Cell and Gene Therapy Forum, we have a group focusing on bioburden and sterility testing, looking at the assays and how we can reduce the impact on batch yield. That is an ongoing program. I will hopefully have an opportunity to flash up all the other topics in a little while, although we've got lots of great questions. But if you're interested in this and you're part of the BioPhorum, please reach out, or just reach out to myself as well afterwards, and we can possibly help. And if not, this publication will be coming. The team are working towards it. They hope to publish on this. So if this is of interest, please stay tuned to the BioPhorum publications. Skipping down from Benson, does the container for reference material need to be the same as the drug product?
The container for reference standard?
The reference material, yes.
Well, I mean, typically containers, any kind of container closure systems, as well as containers that your drug product is stored in, has the opportunity to contribute to any degradation or stability of your material. So I would expect that you would want to have in the same container to ensure that you don't introduce additional stability issues to your reference standards.
Okay, thank you. Now there's just over about 6-7 minutes left, so I'm going to try and get through as many of these final questions as possible. So hopefully you'll be able to help here, Sharif. So has the FDA accepted platform data for CGT programs, as they seem reticent for biologics?
If I'm not mistaken, I know that certain there are, first of all, there's a handful of cell and gene therapy products that have been commercially licensed. But there are in the pipeline clinical programs that are being supported by platform processes within the contract manufacturing network. There are programs that are using platform processes that have been optimized within the CMO, for example. Now, whether the FDA has accepted those, that I couldn't comment on at this time, but for individual companies, I'm aware of that there are companies who have multiple assets that are in development, are utilizing a more standardized platform process within their organization.
Great, and I can also say again, another group within BioPhorum CGT are discussing the platform approaches for validation as well, so that's being discussed. Just trying to look at the last few questions in about 3-4 minutes. Does every unit operation need to be validated for PPQ?
This is pretty much the expectations are the same. We need to understand our processes. We need to understand what are the critical process steps, and the expectations are that you validate critical process steps. So I wouldn't necessarily say whether that every single step requires validation during PPQ, but it is up to us as the manufacturer to decide what we consider to be critical, and then demonstrate reproducibility and consistency in those process steps. So I wouldn't say that one or the other. It's up to you to decide which is critical and validate those.
And I guess following on from that, we have a question: what's the best way to document the use of surrogate as opposed to product? Is a risk assessment?
It's not only a risk assessment, Stephen. I think you also need to, from my personal experience, it's always good to have a standalone document, but that document then can be summarized in your validation protocols as why are you using this and what is your justification. If you have a risk assessment, if you have additional studies, they can all be put together into one document, which can then be used as a reference and as a justification in your validation protocol and your validation final report that can then be presented to an auditor during a PAI or some other additional agency discussions during scientific advice or and so on.
Okay, hopefully, Jenny, that helped with your question on risk assessments. So we've got a couple left, and then I'm going to have to just skip on to keep us in time. Some questions. So here's possibly a little off topic, but doing PQs on cell therapy equipment, which focuses primarily on pump functions, but are others doing this? And I think we're seeing some other people are saying yes, doing others OQ, PQ. So any thoughts on equipment validation here?
What was the question again? To use
Are others doing PQs on cell therapy equipment beyond the vendor IQ, OQ?
Yeah, I think it would be very much dependent on what that piece of equipment is used for. I mean, IQ, OQ is pretty standard, but then depending on, what equipment you're looking at and when you do the PQ, it really is equipment dependent. I don't think there's a general process that you can follow there.
Right, thank you. And I think we've pretty much got through most of those questions with a couple of minutes left. So thank you once again, Sharif, for all of the feedback, comments, and everybody, thank you for all your questions. Now, in the last minute, I want to make sure you try and get to your next meetings. I'm sure you have on the hour. So very briefly, as I alluded to in some of these questions, lots of other conversations are going on within BioPhorum Cell and Gene Therapy Group. So this slide is showing you we have 28 sub-teams. The validation challenges team has now closed, but there are lots of topics going on. I mentioned platform approach is being discussed. I've mentioned that we've got phase appropriate approach to assay validation. We're talking about minimizing the impact of batch yield, where we're doing sterility discussions. So there are lots of topics going on. So if you see anything of interest, please follow up with myself, and I will share my email towards the end, so you'll have that so you can reach out if you are interested. And lastly, thanks again to Sharif, but this is one of a number of series of webinars from the Cell and Gene Therapy group. As you can see here, we have our next webinar is on potency assays from the potency group. And within that, they're going to be talking about how they recommend a matrix approach to potency testing. And here is the link just in the Teams chat. So please click on that to join that one. And then also, as you can see, there is an IT user stories for IT within CGT. That is a further webinar coming up, and I will share the link to that. Sorry, this is going fast towards the end. Lots of great questions. So please follow these links and sign up to these webinars. Hopefully they'll be as interactive and lots of great questions as we had. And lastly, this webinar is recorded. It will be shared with you all, and I hope you've enjoyed it as much as myself. I've enjoyed it. So thank you, Sharif. It was really great. I really appreciate that. And thank you, everybody, for joining, and have a great day. Thank you once again.
Thank you very much. Thank you. And best of luck to everyone as well.
Thank
Take
you.
care.
Bye all.
Bye.